Thank you very nice. Thank you very much for invitation. I will be talking about the research related to eye, and because it's like photonic meeting, I will be focusing on the optical techniques. So we are International Center for Translational Eye Research, and we are part of the Institute of Physical Chemistry, Polish Academy of Science. The center was established within the International Research Agenda program from European Union, and we already received this funding twice. This year we started second phase of this. So before I start going in a little bit of details about the optical parts, let me introduce the center itself. So we have 5 groups. First group, Physical Optics and Biophotonics. It's led by Professor Maciej Wojtkowski. And this group is focused mostly on developing novel imaging modalities. And you can see here the schematic of one of the systems that is, we believe, another generation of optical coherence tomography methods, and colleagues call it STOCK-T. When they acquire images, volumetric images of human eye elements very fast with very good accuracy. So then we have image-guided devices for ophthalmic care group, and I'm acting PI in this group currently. The former PI, Dr. Andrea Culatoro, moved to Milan for the professor position. In this group, we are focusing on to study eye function and also to monitor various eye treatments. Also around eye surgery planning, eye surgery guidance with optics. And at the end, we would like to also focus a little bit on low-cost solution. And I will also say a little bit about this later on. We not only have optical groups like imaging groups or kind of technology groups. Dr. Beto Fernandez is leading integrated structural biology group, and they are working on various proteins involved in the vision cycle and how to use them to improve things, maybe learn something fundamental about the vision process and what's happening in the retina, and also provide some therapies. Another group of this kind is Ophthalmic Biology Laboratory, led by Dr. Andrzej Fojk. And the focus is to work on various mouse models to study the vision both in eyes and brain. And for example, he's trying to use rabies virus as a kind of delivery tool for therapies. And the last group is computational genomic group, and PI here is Marcin Tabaka, and they work do a lot of experimental and computational studies on single cell transcriptome and epigen profiling. So I'm not so familiar on those biology parts, so I will focus on optical. And a lot of activities in the center actually is driven by some of the work of Chris Palczewski, who actually was involved in the first International Research Agenda program that we received. And he's working on the gene therapy for various eye like vision conditions. So he believes that in a decade it will be possible to cure a lot of them with these new technologies. And because those technologies are approaching, it's important to do a lot of things around them. So let's say how to deliver the treatment to this specific regions of the retina, and how to define to which region we should inject this gene, and also to study not only the structure of the eye, but also the function. So let's go back to POB group, and the group developed. The method was developed over almost a decade now, I think. And it's called spatial-temporal optical coherence tomography, and it's kind of holographic OCT. So the images are acquired in full field scheme, So all pixels are acquired at the same time. It's demanding for the camera, and I will mention this at the end, and it requires slightly different light sources that we normally use for scanning spot OCT. As you can see, they can really collect very nice high-resolution images of the retina and also of the cornea. And they use some tricks to play with coherence of the light that is used in the method. For instance, this helps to reduce crosstalk from the deeper layer, which is usually drawback of such techniques. So they show that with this crosstalk reduction, they can really, I think, probably for the first time, image the choroid layers and then study choroid capillaries in detail. Because the technique is very fast and it's acquired in kind of full field fashion, we have very stable phase along whole plane of that we are imaging, and that allow us to use phase sensitive methods. For instance, you can apply some stimulus to the eye, like in this case, colleagues applied flicker stimulus with this kind of stripes, and you can see on the left that we are able to recover these tiny changes. Those light-induced changes in the retinal layers are at the level of few or tens of nanometers, which is we can measure very tiny changes in the living eye. And all this, like this functional imaging, will also be important to study whatever those gene therapies are really doing what they should do with the retina in the mission of preserving the vision. We also have some fancy things that were probably, I believe some of you will not recall that something like this is possible. In terms of that, we can see infrared light in specific condition. If we have laser that is pulse laser, and I think in this case, you need at least nanosecond pulses in the range of 900 to 1200 nanometers. You can perceive light that will be roughly equivalent of second, like two-photon effect. So let's say it started when we've been using 1060 swept laser for retina imaging, and when we align the patient, we usually scan the eye in the kind of cross fashion, and we perceive the green cross, which is kind of not intuitive why we see it. And then Kasia Komar, who is leading this part, did a lot of work to really study how this works for different wavelength in this region. And it's very promising because it looks like that this technique might be more sensitive to study some kind of quality of vision than equivalent studies done in visible range. So they are still exploring this. And there are some other aspects that I cannot mention yet because they are kind of patent pending. But hopefully in future we'll be able to share it. The other part with this two-photon process is that we can, my colleagues show for the first time in humans that you can use two-photon process to do kind of laser scanning of ophthalmoscope. So they kind of develop the system. The signal is pretty low, but you can do averaging of multiple frames that are aligned based on classical techniques. And this technique is really explored with our group and also Chris Palczewski and his wife, Grażyna. So for IDOC, Interoperative Devices Ophthalmic Care Group, part of the work is to add some optical components to surgical kind of scenarios. In this case, what we can see is the screen of the combined panel. We see the view of the standard view of the surgical microscope on the top left. So you see artificial eye and the needle. Close to it, you see two grayscale images. These are OCT images acquired. One is along the needle and the other is perpendicular to the needle. And the idea is here to track the needle and display those preview images. So those will give you additional depth information for the surgeon, which is missing in the classical approaches. And you can track the needle by vision means, so we can acquire images from the camera and do some kind of on-fly segmentation of the needle, maybe with machine learning. Or in this case, we decide to place the tool with the needle on the wrist of the robotic arm. And if everything is calibrated well, we can send the information from the robot about the position of the end of the surgical tool to the OCT software. And on the fly, in this case, it's 200 Hertz refreshment rate. We can update the scanner pattern that is used to scan this B-scans along and perpendicular to the tool. So this is our fresh result that we are drafting the paper on. And the other example on this, and it's also close to the clinic, there is this technique for tonometry measurements when you apply air pulses to the eye. And probably some of you may have those measurements done in the ophthalmic clinic. And then you have infrared light illuminating the cornea, and you measure the amount of light that is reflected from the cornea's surface. So this is in this kind of standard clinical measurements. However, in this case, you do this measurement indirectly. 10 years ago, we proposed to combine this air path stimulation with the OCT, so we have direct measurement. And we did it only for the central point at the cornea. Of course, the cornea pathologies, for instance, like keratoconus, they don't occur central symmetric. So we'd like to probe more points. We did some work with Andrea Curatolo on the multiple meridian approach that you can acquire those corneal deformation at 1 kilohertz. But I believe that you need better temporal resolution because the process of deformation recovery is like around 20 milliseconds. So what we developed here, and you see on the left clinical prototype that went to clinic and measured size, more than 100 eyes already. We have multiple spots distributed along the cornea surface, along main meridians. And with current OCT technology and light source technology and digitizers, we can acquire all those points simultaneously simply by adding some phase delay to each channel. And in the middle, you can see an exemplary image of such deformation measured at 9 spots simultaneously. And just to give you some hint, we are working on big paper on this, so I cannot share full details. From this clinical measurement, we can show that some parameters extracted from these deformations give us ability to nicely separate a control group from a keratoconus group. So this looks very promising. And here we hope that in future we'll be able to translate this device to more advanced product that will be possibly handheld, and this will require some additional work, some switching from bulk optics to maybe fiber bundles or something like this. And we'd like to also lower the device of the cost. So because this is like a photonic industry meeting, I decided to add additional slide that will give you some hints about what kind of tools we are using in this optical research around the AI. So we have two paths. One is on the left. We develop novel system, or we focus on basic science, and then the price tag of the components is not so important for us. If it's expensive, yeah, we like to have it like, get it cheaper with some discounts, but we can live with this. And on the other hand, we have more like product-driven approach. And usually when you do some market analysis, you end up with this information that you should move toward low cost. And it was actually the market for this 9-spot approach that I showed you on previous slide. And then also some small footprint solution. So on both we have light sources. For this 2-photon, we use pulse lasers, so nanoseconds to femtosecond regime. For OCT, we use swept lasers. And for standard OCT, let's say we use lasers that tune the wavelength with more than 100 kilohertz repetition rates. Bandwidth depends on the spectral range. It can be around 100 nanometers, and ideally output power should be around 20-30 milliwatts or more. For this full field OCT, so stock T approach that I show you already, we go to the slightly different approach. We use slow laser, so it's sweeping around 1 kilohertz. The bandwidth will be from 75 to 100 nanometers. Also depends on the central wavelength. It can be 800 or 1060. And in this case, we would like to have powerful. So probably if you can deliver something that is more than 50 milliwatts, we will be happy. And perfectly, it will be sweeping linear in wave numbers. On the right hand, so this more like product market stuff, we still can use the standard OCT lasers, but I gray them out, so maybe it's not so important approach. But if you want to go to low cost, the approach is to slightly lower the specs of the laser, so we can, let's say, reduce the repetition rate to a few kilohertz. We can shrink the bandwidth, and then still we'd like to have few milliwatts of optical power. And the current approach is with this, if you use VCSEL diodes with current tuning, so there is kind of internal heating process that will tune the wavelength. And then on detector sides, with these novel systems, or when we do basic approaches, we have digitizers that work in gigahertz range. In some cases, we require nonlinear sampling, so like K-clock. The example of this company that produce such digitizers is Al-Azhar. And these are pretty pricey. So usually it's second most expensive element after the light source. And we have ultrafast for this full field OCT. We use ultrafast cameras like Proton company. These are very expensive. They are not designed for science. And basically, I haven't seen solutions that are outside visible range. So for some cases, we like to have like infrared, like 1060 or around. Detectors on the right side, because we have this low spec laser, and if we look at those simple equation in OCT for resolution and maximum range that you can measure with OCT, you can kind of easily notice that for a low spec laser, we can end up in a low spec digitizer. So we can use basically lower sampling frequency, and then you don't have to pay like 70K euro for the digitizer, but maybe a few hundred euro or dollars. So this is kind of space that we can move on. I think what is marked in the red, so this the slow laser with this pretty broadened bandwidth of tuning, it's really important because it's for this new generation of OCT. I think this is the future of this retina measurements. Maybe pharmacy will use it to study the drug developments and so on and so on. And actually, it was a big problem with those because these were produced by Russian company. I mean, not Russian, the company that used some Russian components. Part of this company is based in Ireland, but the key components were produced in Russia. So for a long time, there was no access to those sources. We had 2 or 3 of them, but you know, if you want to develop new devices or something is get broken, you are done. So this is really interesting space for industry to catch up because I think there is future, and also in this low-cost VCSEL tuning diodes for low-cost products. So I will stay with this nice photo of ICTER family. If it was kind of, you know, introduction, I went through those ideas very briefly. If you are interested, we can explore specific topics. So we can look at ICTER website, or you can email me and we can maybe discuss things in details later on. Thank you.